Okay, so let's talk about the eukaryotic cell. First and foremost, what organisms are considered eukaryotic? Meaning they're made up of eukaryotic cells. They could be a single eukaryotic cell or they could be multicellular. Now, I'll remind you the multicellular eukaryotic organisms are not considered microbes. So, for example, animals and plants are multicellular, they're big, so they're not typically studied in a microbiology class. What would be? Well, fungi. So yeast cells, for example, are eukaryotic. And that's why, if you think about it, antibiotics are given for bacteria, but they're not given for yeast infections. And that's because typical antibiotics target things in a prokaryotic cell that are not found in eukaryotic cells or that are different in eukaryotic cells. But a yeast or a fungal infection, that's a eukaryotic organism. Protozoans are eukaryotic, so like the parasite that causes malaria or trichomonas. Algae, even though again algae doesn't cause human infection, that's a eukaryotic cell. So what would a eukaryotic cell look like? Well, these two drawings um, show the typical features of eukaryotic cells. This would kind of represent an animal cell. This would represent a plant cell. You'll notice they seem to have a lot more going on the inside. So they've got a lot of membrane-bound organelles. The nucleus, which contains the chromosomes. It's got mitochondria here in red, which generate a lot of the energy for the cell. There's what's known as the endoplasmic reticulum, which we'll talk about. The Golgi apparatus, which is found in eukaryotic cells. So there just happens to be a lot more complexity to eukaryotic cells. So let's talk about some of these organelles. First and foremost, mitochondria. Mitochondria are known for generating ATP. ATP is the cell's primary energy currency, meaning if it wants to do something that requires energy, it needs ATP. Now, do prokaryotic cells produce ATP? Yes. They just don't have an organelle that contains all these enzymes. They actually have a lot of the same enzymes, but they don't have it all in a single organelle. So mitochondria are the site of aerobic cellular respiration. That's the fancy scientific name for generating energy, but it requires oxygen. So oxygen is aerobic. It also is going to require some form of energy like glucose or an amino acid. So the oxygen just helps the process occur. Oxygen doesn't actually have energy in it. So mitochondria typically looks like this. It's got two membranes, an outer membrane and this inner membrane, but it's got a bunch of enzymes that allow that cell to generate large amounts of ATP. Chloroplasts. So chloroplasts are the site of photosynthesis. So organisms that can extract energy from sunlight are going to require chloroplasts. Or some bacteria can generate energy from sun. So some bacteria can photosynthesize. They're called cyanobacteria. They don't have chloroplasts, but they have a lot of the same enzymes. So they can still undergo photosynthesis. And I'll remind you what photosynthesis is. It's utilizing sunlight, water to produce a carbohydrate or sugar, and then you need to get energy from that sugar. So if you look at the plant cell, for example, they have chloroplasts. They also have mitochondria, whereas an animal cell won't have the chloroplasts. An interesting theory to talk about. It's called the endosymbiotic theory. There is strong evidence that mitochondria and chloroplasts were once bacteria. So what happened a long time ago, when we had these ancestral prokaryotic cells on Earth, they started to become more complex and they started to form organelles. So they started to bring in their plasma membrane and then surround the DNA with that plasma membrane, which formed a nucleus. Then what's believed is there was some other prokaryotic organism that got taken up by this cell and it formed a mitochondrion. And that kind of began the lineage that led to an animal cell. Plant cells took up these other prokaryotic cells that could photosynthesize, and that became a chloroplast. So now you got a plant cell that had both mitochondria and chloroplasts that originated from other prokaryotic cells. So this is evolution. So you kind of have to believe in the fact that cells evolved in order to form more complex cells and then in order to form multicellular organisms. But this is a pretty strong belief in the scientific community. Now, how do they know? Well, one thing is there's genes in mitochondria and chloroplasts. So mitochondria and chloroplasts have DNA in them. And a lot of the genes that are in mitochondria and chloroplasts are very similar to the genes that are in certain bacteria on Earth today. So it believed that, some of these guys didn't get taken up and they eventually, stayed on Earth and they're still on Earth. They evolved a little bit, but there are some bacteria that have genes just like the genes that are in mitochondria. So it's pretty strong evidence that that's what occurred. All right, next organelle, the ER, or endoplasmic reticulum. There's two types, or two parts of it, I should say. One part has ribosomes. So these little dots represent the ribosomes. So it's known as the rough ER. So basically, when they first kind of were able to take really high-powered pictures, they found that parts of the ER had these dots, so it looked rough, whereas other parts didn't have the dots, so it looked smooth. So that's how they came up with the two names. Just so happens the function of the two is different. The rough ER, because it has ribosomes, is involved in protein synthesis. The smooth ER doesn't have ribosomes, but instead it synthesizes lipids. It also is a site of degradation, so it can actually kind of break down things for the cell. And then it also can store calcium. This is important for our cells, like muscle cells. Muscle cells have to store calcium in the smooth ER. Golgi apparatus is going to get a lot from the ER. So the ER is going to produce proteins and lipids, package them in these little vesicles, but then the little vesicles will go from the ER to the Golgi, and then the Golgi does some modifications to it. So it modifies some of those proteins, it modifies some of those lipids, and then transports them out to their final destination. So a lot of times we think of the Golgi as kind of like a modifying, packaging, and shipping site. So you kind of think of it as maybe like a UPS store where you bring stuff there, you bring your proteins, you bring your lipids. They do a little modification, right? They'll package it in a box, maybe wrap it for you, and then they ship it out to the final destination. So that's the job of the Golgi apparatus. A couple last organelles, lysosomes. They also kind of break things down for the cell. So the cell can it's kind of like a recycling facility for the cell. The cell can, if something gets worn out or used up, we can send it to the lysosome, break it down, reuse the parts to build a new, protein or a new organelle. It's very useful for certain immune cells. So when we get to the immune system, we'll learn about phagocytosis. That's where we have certain immune cells, like our macrophages and our neutrophils, which can phagocytose bacteria, so basically bring in a bacterium, and then they fuse it with the lysosome. And the lysosome uses these enzymes to degrade and break down the bacteria, in essence, killing the bacteria. So lysosomes are important for immune cells. And then peroxisomes, you don't really hear too much about those, but they do a number of things for cells. They can detoxify. So, like your liver has a lot of peroxisomes because your liver is a major detoxifier. But what they're named after is the fact that they generate hydrogen peroxide. They generate that as kind of a byproduct of what they're doing. So, those are the organelles of the eukaryotic cell. This slide and the next slide you'll see is a big, huge table, but I wouldn't worry about every little thing on this table, but it's a good kind of table to look at descriptions for each of these structures that eukaryotic cells have. So I'm not going to go through it because it would take up another 5-10 minutes. But you can see, you can go through and look at these structures and read the little description. And most of these we covered on the previous few slides. This slide kind of takes the eukaryotic structures on one side, describes them, and compares that to prokaryotic. So this is kind of a big summary of prokaryotic versus eukaryotic. Anywhere from the size comparison, how they divide, and then comparing some of the structures. This is a little bit too much information for us to focus on. So I would just go back and look at each of those slides, make sure you understand what each of the organelles do for that eukaryotic cell, and note whether that structure is in a prokaryotic cell as well, like a ribosome. They both have ribosomes, or if it's exclusive to the eukaryotic cells.